Thank you, Daria and Kevin, and good afternoon, everyone. It's a great pleasure to be here and celebrate this important milestone of the SPARK program. So as Kevin mentioned, and I think the last several years really underscore the huge unmet clinical need for development of novel antiviral strategies. So the current approach focuses on targeting viral enzymatic function. This approach has shown great success for the treatment of chronic viral infections such as HIV or hepatitis C virus. However, this approach to drug development really does not scale easily. It takes on average 12 years and over $2 billion to move from the identification of viral enzymatic function all the way to an approved drug. And the spectrum of coverage that achieved with these direct-acting antivirals is typically narrow, ranging from a single viral genotype to few related viruses at most. So overall, this makes the antiviral development quite slow and expensive, which ultimately limits drug access. This is an impractical approach for developing antivirals for emerging viruses. This is a large number of viruses for which we, at most part, don't have vaccines or antivirals, including viruses such as dengue, Ebola, and Zika. Since we cannot predict future threats, this approach does not provide global threat health protection and no national security readiness. And lastly, when you use these direct-acting antivirals as monotherapy, there's high likelihood that resistance will emerge. So the approach that we propose instead is to combine these strategies with host-targeted antiviral approaches. As you know, viruses replicate inside a host cell, and thereby they co-opt host cell machineries for every step of the viral life cycle. So if we have a way to identify host functions that are required by multiple viruses, then these could represent potential targets for broad-spectrum antivirals. We can then go on to discover new chemical entities that target these targets, or perhaps even more attractively, repurpose already approved drugs that target these host functions. So overall, this could reduce the cost of drug development and create potentially a faster path to clinic, can provide readiness for future outbreaks, and such a broad-spectrum antiviral can potentially be administered prior to the accurate diagnosis of a viral pathogen. Since such approaches are not under the direct genetic control of a virus, the likelihood that resistance will emerge is also lower. So my research program focuses on better understanding the interactions between viruses and the host, and we've been focusing quite a bit on how viruses traffic inside the cell. Specifically to this project, we have been studying the roles of a group of adaptor protein complexes. There are 5 such complexes in mammalian cells, and each is known to sort in a distinct intracellular pathway. Shown here with the schematics of the hepatitis C viral life cycle, we've shown that viruses hijack these adaptor proteins to mediate distinct steps in their life cycle. In the case of HCV, we've demonstrated a requirement for the AP2 adapter, which is an endocytic adapter in entry, as well as in viral assembly via an interaction with the capsid protein. We've shown that the AP1 adapter, which sorts in the secretory pathway, is hijacked by yet another viral protein, a non-structural 2 protein, to mediate later steps in the viral life cycle, namely release and cell-cell spread. Using live cell microscopy, we've shown that these are indeed the buses, if you will, that HCV uses to traffic inside the cells. So here you're looking at individual viral particles labeled green, and essentially green goes together with red, which represents the individual adaptors. We can quantify these viral particles, and we've shown significant co-trafficking with the individual adapters, unlike a control protein. And importantly, conditions that disrupt those virus-host interactions that we've identified, in this case, a mutation in the HCV capsid protein, disrupt this co-trafficking. Importantly, there are 2 host cell kinases named ACK1 and GAC. These are known to phosphorylate the mu subunit of these heterotetrameric adapter protein complexes and thereby stimulate their binding to host cargo proteins. What we've shown is that ACK and GAC similarly regulate the virus-host interactions that we've identified, and thereby they regulate distinct steps in the HCV life cycle, namely entry, assembly, release, and cell-cell spread. They can therefore be viewed as master regulators of viral infection. And importantly, we have validated the requirement for these kinases, for the life cycle of multiple unrelated viruses, suggesting that these kinases may represent attractive targets for broad-spectrum antivirals. We then looked at affinity assays and heat maps of already approved kinase inhibitors and identified a group of compounds, including sunitinib and erlotinib. These are two anti-cancer drugs, which have very potent activity against ACK1 and GAC, even though ACK1 and GAC are not their cancer targets. We've demonstrated in vitro activity with these drugs and showed that they inhibit all those distinct steps of the life cycle which are regulated by ACK1 and GAC, namely entry, assembly, release, and cell-cell spread, and really have no effect on HCV RNA replication, which is the step of the viral life cycle currently targeted by approved direct-acting antivirals. We've since demonstrated activity against a large number of viruses, including some very important clinically relevant viruses. From the Flaviviridae family, we showed activity against the 4 dengue serotypes, against West Nile, as well as more recently Zika. We've shown activity against Ebola and chikungunya, as well as additional viruses. And importantly, we've demonstrated that the combination of the two drugs generates a significant synergistic antiviral effect against a large number of these viruses. We then wish to determine whether dengue can escape treatment with these drugs. So to do so, we passage cells that are infected with dengue over many weeks. And we use as a control a direct-acting antiviral shown here in red. And what you can see with this control is that there was a reduced level of infection. Nevertheless, at some point there was rebound, which coincided with the emergence of previously characterized mutation to this NS4B inhibitor. In contrast, as you can see in the blue curve, the combination of these two drugs continued to suppress infection over many weeks, suggesting that the genetic barrier to resistance to this host-targeted approach is higher. Moving to in vivo data, here we use dengue-infected mice. These are AG129. This is a well-characterized dengue model. These mice are knockouts for interferon alpha, beta, and gamma. We inoculated them with dengue and started treatment upon inoculation for a duration of 5 days. Here you can see the effect of the combination of the two drugs on viral load in the serum or viremia. And you can see that between day 2 and 3, when viremia continues to increase in the presence of vehicle, the treated arm suppresses viral replication by over a log. Monitoring weight loss in these animals, you could see that mice that were treated with a vehicle continued to lose weight, whereas mice on treatment lost weight, but then started gaining weight and essentially recovered. And here's the mortality data, whether the drug was given intraperitoneally or orally. We saw substantial protection for mortality in these mice, whereas the control mice succumbed to infection on day 4 to 8. Importantly, this protective effect was also demonstrated when drug was administered at various time points post-infection. So this is a much more biologically relevant model. And you can see that mice that got the drug even after 48 hours, the treatment delayed death. However, this group eventually succumbed to infection. But this suggests that this strategy could be used not only as a prophylactic treatment, but also for post-exposure. Moving on to yet another disease model. These are mice infected with Ebola. These experiments were done by my collaborator, John Dye at US Emory in the military. Here, B6 mice are being used and animals are being inoculated. Treatment is initiated upon inoculation for a duration of 10 days. And you can see that while the individual drugs protected mice to some degree or had no protection, the combination of the two drugs caused protected about 50% of these mice. And again, those mice that recovered essentially gained weight and behaved normally. So where is this project standing as far as clinical development? During the Ebola outbreak, this was selected by Gates Foundation as one arm out of 3 of multiple treatments for Ebola virus disease study. By the time everything was ready for the study to launch, the outbreak essentially resolved, but this is now being viewed as an approved in-drawer protocol. For the dengue indication, this was licensed by a small biopharmaceutical company named Globovir Biosciences, and we hope to conduct a pilot study. FDA has already reviewed this project, and we are currently addressing some of their comments. There's also a possibility of conducting a study elsewhere under foreign approval. Nothing of this has been made possible without of SPARK. There were many challenges along the way, both during the preclinical phase as well as the clinical phase. Just to name a few, SPARK was very instrumental about helping us to choose disease models to focus on, provided help with target validation, preparing drug formulations for in vivo studies, designing the in vivo efficacy studies, PK studies, and more recently continue to provide a lot of medchem support for the development of selective inhibitors of these targets. In the clinical phase, you can only imagine how challenging this was both politically and logistically. The amount of support that was provided, particularly by Kevin, who joined me to USMREAD and knocking on many doors, was tremendous. So thank you again for all this. And again, thanks for all the SPARK consultants. Happy anniversary. It was really a great privilege to be part of this program from the very beginning, initially as a postdoctoral fellow at Jeffrey Glenn's lab, among the first round of SPARKees, and then all the way when it was just still a small spark, maybe even experimental adventure, to where it is today, so full sky of fireworks. So it has been really a pleasure, so thank you all, and I'll stop here.
So we have time for a couple of questions.
Yeah, I'm so surprised you took same strategy with us. So we already developed host kinase suppressor as an antivirus drug. And now it's in the clinical trial in our hospital. But we are targeting DNA virus, not the RNA virus.
Okay.
So my question is, your drug is applicable for DNA virus, just for RNA virus?
So thus far, none of the DNA viruses we've tested have been susceptible to these drugs. We're still not quite know why. Perhaps they do not require these kinases, but the majority of RNA viruses that we've been testing are susceptible. So,
thank you.
Sure. Thank you.
The preceding program is copyrighted by the Board of Trustees of the Leland Stanford Junior University. Please visit us at med.stanford.edu.